Thank you very much, Jessica, for the introduction. I'd really like to thank Sarah Davidson for inviting me to the seminar series. I know that many of you have seen a few Rust presentations, and so I'm happy to be here, and I hope that you'll learn a little bit more even about UG99. And so it's really a joy to be here, and I thank many of you for meeting with me and for the discussions that we've had. And so in this presentation, I'm really going to break it into two different components. And so first, I'm going to try to answer the question, what is UG99? And this is the introduction to the talk. And then after this, I'm going to explain a few of the things that we're doing. And I'm going to put this in the context of what the community is doing, but also really narrow in and what my lab specifically is doing about UG99. And so this picture here shows a farmer in a wheat field in Kenya, and these pictures were taken in 2007, and this is the result of a UG99 stem rust epidemic. And so you can see in the field in the bottom right a lot of lodging of the stems, and in these pictures that are zoomed in to the wheat stems, you see this dark brown uridinospores on the stems. And so this weakens the stems, it causes lodging. And then also within the field where you see the farmer standing, you can, if you just take a quick glance at it, you may not think that there's anything wrong with that field. But if you were to go to those spikes and look at the seeds that are inside, you see very small shriveled seeds. And so this is really the problem with stem rust, is you're reducing the yield. And so farmers went out into their fields in Kenya in the mid-2000s and went out with a combine and really didn't harvest anything because it was all just blowing out of the end of the combine because the seeds were so light. And so we know that UG99 is a very devastating disease when it gets to epidemic levels. But to really explain more specifically what it is, it's a fungal disease caused by Puccinia graminis. And the way that we understand this disease is by classifying it according to its race phenotype. And to explain what that is, I'd like to first show this picture here. And what this is a slide of seedling infection types of wheat leaves. And they're displayed on the 0 to 4 scale, which is a qualitative scale that helps us to look at resistance and susceptibility, but more specifically for looking at the pathogen, avirulence and virulence. And so the seedling infection types ranging from 0 to 2+ we consider as low infection types, and the 3 and 4 we consider as high infection types. And when we take an unknown sample of rust from the field and purify it as a single spore culture, we evaluate it on this list of wheat varieties here. And so I think I'm just going to walk over and point at the slide. The laser tends to make a noise when I use it. So this is the name of different wheat lines here. And in this column is the stem rust resistance SR gene that those different lines contain. And when we evaluate those lines with an unknown isolate of rust, you see the result that we see. So you see these 4 up here, these 4 lines are all susceptible, and the seedling infection types correspond to that. A 3 or 4 is susceptible. This is the isolate name for this particular isolate, so 04, 2004 Kenya, and then the number. And then you see there's only a few lines that are actually resistant. So you see this SR36 is very effective to this isolate, also SRTMP. And in this set, what we can do is actually classify the race phenotype. And so we have this nomenclature system that I'm going to talk about for a minute or so, just because I think it's important for the rest of the presentation. And so the race phenotype of this isolate is TTKS. And so the isolate name is the 04 Kenya 156, and the race name is TTKS. And we come up with this race name based off of the reaction to these differentials. And basically the code is shown here, where we see high as the reaction, and according to this code, we can find the letter that it corresponds to, in this case, a T. So that's where we get TTKS. The significant thing about UG99 is that it's not just one race. It's several different races. And so, again, in this slide, you can see that there's a few different isolates that we have, 3 different isolates, and those isolates have different race phenotypes. So this one from 2007 in Kenya is actually high on SR36. And then there's this one collected in 2006 is high in SR24. So it's not just a single race, it's a group of races, and that's what I think we can consider the commonly used term as UG99. The other thing I think that you can kind of pick out right away is that UG99 is defeating almost all of the genes that are in this differential set. And so that's one of the clues that this is actually a very dangerous race. And then to also kind of clarify, in this slide, we added a 5th set. And so there's 5 different letters now. And that was to help discriminate some of the new races of UG99 that we have. These new races are very significant because they knock out additional genes that are very important for agriculture. And so these genes were, especially SR24 was widely deployed in Kenya right after UG99 was found, but the pathogen adapted to that deployment of a resistance gene and broke it down. So not only is it broadly virulent, but it's also adapting to overcome the resistance that's used. And when you look at a graph of where UG99 is, which is displayed here, you can kind of see its distribution and also the composition of the different races. And so I've listed a few of the race phenotypes in different colors. And so you can see that the original TTKSK is common in 2012 in this figure throughout Eastern Africa, but different members of the UG99 race group are common in different areas. And so it's not just a single race, it's kind of a mixture. And then the gray is not UG99, those are other races. And then another really significant piece of information about UG99 is that it's considered to be a very big threat to wheat production. And the reason why people consider it a threat is from what we know about UG99, what we know about stem rest in the past. And so this is a graph which is showing on the x-axis year starting in 1918 and going to 2008. And on the y-axis is the percent of the total US crop that was lost to stem rest. And so you can see that during epidemic years, it was not uncommon to get yield losses across the entire United States of 5 to 10%. And in one particular year, 1935, almost 25% of the whole United States crop was lost to this disease. And so from the past, we know that stem rest was a really big problem. And when you look at what happened during that epidemic year in 1935, the results are kind of broken down in this figure by state. And so you can see that the yield loss estimates for different states. And in 1935, in the Dakotas and Minnesota, 50% of the wheat crop was lost. So this is a huge problem, though 25% of the nation's crop was lost in a particular state or even in a region, you can see it can be a very devastating disease. And many of you know that wheat is broken into different market classes, and so this is almost annihilating like a particular market class of wheat for the United States. So it's very scary when we think about what happened in the past. So in 1935, that epidemic was caused by a particular race of stem rest. And also in the 1950s, there was another race that came in and caused epidemics. And so what we've seen in the past is that people have deployed varieties that are resistant to stem rest, and those varieties have different resistance genes. Those resistance genes are overcome by the pathogen, and then you get an epidemic. And then new resistance genes are deployed in new varieties. Those are grown, and then the pathogen adapts to those, and you get a big epidemic and yield loss. What's very, I think, underscores the work of a lot of scientists in the last century is that there hasn't been a lot of yield loss in the past 50 to 60 years. And so there's been a really big emphasis on stem rust resistance breeding, and that's really eliminated this as a major threat to US production. But unfortunately, with UG99 being detected, we see that in the United States, the majority of our currently grown wheat cultivars are susceptible. So if UG99 were to arrive in the United States, we might be back in the situation we were in the 50s or 30s, where if the conditions are right, we might get a huge epidemic. And this graph just kind of shows the proportion of varieties in different regions of the United States that are susceptible or resistant. And so you can see that most of the varieties are susceptible to the original TTKSK, and some of the varieties are susceptible to some of the new variants. And so this really puts us in a very vulnerable position, which is why I was hired by the USDA to work on a disease that's not even in the country yet, which I think is kind of indicative of how much people are worrying about this. There's been a little bit of modeling of what UG99 might do, and this is just a heat chart of showing where wheat is grown in the United States. And if you compare that to historical stem rust losses in the United States in the second chart down at the bottom, and kind of multiply those two layers together, you get this chart, which is kind of an estimated perfect storm of where UG99 would cause the most problems. And so it's definitely a disease that we think would mostly impact the DNS wheat, the dark northern spring, high quality wheat in the north. And so I think it's a very important disease that it could be quite devastating if it arrives in the US. So in summary, I hope I've kind of shed some light as to what is UG99. We know that it's first identified in Uganda in 1998, and that was characterized in 1999, and that's where we get the name UG99. But also, it's not just a single race or a single isolate. It's a race complex of Puccinia graminis. And so it's kind of a little bit hard as a breeder to breed against something that's not simple. And the last thing is that it's a threat to wheat production. So moving on to the really cool, the body of the talk, I'm going to explain about what our lab is doing about UG99. And obviously, this is a problem that's a lot bigger than just a single lab working on this disease. So I'm going to start by giving an overview of what the global community is doing about this. And obviously, I think by the number of Rust talks you've had here, I think that you understand the importance of this. So briefly, there's two different broad categories of mitigation for UG99, fungicides and genetic resistance. And definitely the use of fungicides is very key to controlling this disease right now. So in East Africa, big farms that have the resources are spraying 4 times a year or more to control this disease. And so the use of fungicides is really critical. I'm not going to talk about fungicides. I'm going to talk about genetic resistance in the rest of this talk. And so the Norman Borlaug was in Kenya in 2004 and saw an epidemic of stem rust. And he was really key in advocating that UG99 is a big problem. And he formed this Global Rust Initiative, which was renamed the Borlaug Global Rust Initiative. And within this Rust Initiative, there's a number of different projects that are working on UG99. And so here's just a few of the key funders of UG99 research: the USDA, Ag Canada, and the Durable Rust Resistance in Wheat Project, which is based here in Cornell. And so in 2008, the first phase of this DRRW project was initiated here at Cornell, and that was a $26.8 million grant. And then 3 years later, there was another grant for the second phase of that project of $40 million being funded by the Gates Foundation and also the UK International Development Agency. And this slide just shows some of the collaborators on the DRRW project. You can see that there's a number of collaborators across the globe, and really, really key are some phenotyping facilities in Eastern Africa where a lot of the field evaluation can take place. The DRRW project is really multifaceted, and so here's a slide from Sarah showing the different aspects of the DRRW project. You can see that it's not just about gene discovery and marker development. There's initiatives regarding pathogen surveillance and incorporating gender into the project, breeding, seed multiplication. And with regards to my lab, I really specialize in gene discovery and marker development. And so though the project is very comprehensive, I'm just going to really talk about some pre-breeding aspects for the rest of the talk. Here's a facility that we use in Minnesota. So we have a biocontainment level 3 laboratory, which it's a lab that all of the air is contained within the facility. And to get from the outside to inside this greenhouse, you have to go through 9 different doors, all under negative pressure. And on the way out, you have to take a shower. And so anything you bring in stays in or is autoclaved, and you have to shower out. So it's a very unique facility. I think there's only 3 other plant pathogen level 3 labs in the US. And this allows us to work with UG99 in Minnesota. And so we have an assay where we can evaluate material at the seedling stage to make progress without having to go to Kenya to evaluate lines. And here's a picture of this greenhouse in January. So you can see in the foreground a lot of wheat lines that are being evaluated, and in the background you can see Minnesota in January and the snow. But we actually only work in this facility starting in November through the end of March, and that kind of serves as a secondary level of containment. So if worst case scenario, a plane crashed into this greenhouse, it would be too cold for the rust spores to survive. And so we're very confident that we're not introducing UG99 into the US, but it's a very sensitive issue, obviously. Here's a slide showing the number of data points that our lab has generated. And this is just from 2012. We were able to get phenotypes on over 40,000 different wheat accessions or wheat members of populations. And so I've talked with many of you about like high throughput phenotyping, and though this phenotyping isn't very complex or sophisticated, it's very high throughput. And so we're able to generate a lot of data, which has been quite useful in mapping and also in screening germplasm for breeders. And before I really jump into our projects, I'd like to show this slide as the last bit of kind of an overview to the research that we're doing. And so in this slide, you have the different wheat chromosomes, and they're organized by the different wheat genomes. And so in bred wheat, there are 3 different genomes, the A, B, and D, and then the different chromosomes in each of those genomes. And this is a cartoon showing where the known UG99 resistance genes are located in 2008 anyways. And some of those genes are derived from alien relatives of wheat, so wild relatives of wheat that are very resistant, but when you bring those genes into wheat, they come in with like half a chromosome arm or a piece of a chromosome that usually has linkage drag, and it's really not useful for breeding. And what's significant about the slide is you see there really aren't that many genes. And many of these genes aren't useful because they have this linkage drag with them. The red ones are alien genes, and then the black ones are genes that are native to bred wheat, or very closely related to bred wheat. Since UG99 has already shown to adapt to these new resistance genes that are deployed, if you think about we only have a handful of genes, if we put those out, we don't know if that's going to be enough or not. So there's a huge need for additional genes, even though we already have a few that are known. And so to describe the work that I've done in our lab, I'm going to give an overview of 3 different projects. And so those projects are listed here. And so I'm going to just kind of give an overview of those, and we have a lot more than 3 projects, but I think it gives you a taste of the different things that we're doing. And so first I'm going to talk about the identification of a gene in Triticum monococcum. And Triticum is a wheat relative that's related to the A genome of wheat. And so Triticum monococcum is very useful because genes in Triticum monococcum can be moved into bread wheat with a lot of ease. So this slide shows different species of wheat and their genome composition. And so for Triticum dicocoides, Tauschii, Urartu, and monococcum, these are considered in the primary gene pool because they have genomes that are very compatible with the A, B, and D genomes of wheat, and those genes can be moved in very easily. And down at the bottom are more distantly related species that are more difficult to bring the genes in. In this column are the number of accessions that are in the USDA germplasm bank in Aberdeen. And one of the first things that we did was to screen all of those accessions with UG99 just to get an idea of the level of resistance that was out there. We found that there was quite a bit of resistance to UG99, so here's the percentage of those accessions that were resistant. But when we screened those accessions with a couple other virulent races, we found that very few of the accessions were resistant to more than just one race. So really only Agelapis tauschii and Triticum monococcum had any accessions that were resistant to more than one race, with the exception of these very distantly related species, which are very difficult to move genes in. So we decided early on we need to prioritize Triticum monococcum and Agelapis tauschii as sources of new resistance genes. And so for these wild relatives, you can do a seedling assay, just like I showed at the beginning of the infection type scale. And so this is Triticum monococcum seedling leaves that are inoculated with the stem rust pathogen, and you can see that we can discriminate resistance and susceptibility. And within 21 days, we can plant a seed and get a phenotype, which is quite useful for phenotyping. When we look at different accessions of these wild species across different isolates of the pathogen, we can start to see different patterns of resistance and susceptibility that is useful for predicting what genes are there or what accessions might have novel genes. And so in this table, you can see some of the accessions of Triticum monococcum and then their seedling infection types to a number of races of the stem rust pathogen. In Triticum monococcum, there are some known resistance genes that were already in this germplasm, like SR21 and SR35. And since we already know those infection type patterns, when we see something that looks maybe a little bit unique, we can prioritize those lines as sources of potentially novel resistance. So after the initial screening, we selected several accessions to make populations, and here's just one of those populations in Triticum monococcum. And you can see the parents up here, and the resistant parent showing this kind of intermediate response. And then in the progeny, you see some that have the resistance gene and some that don't. And so for this particular gene, it was inherited as a single recessive gene. And what's really nice about having multiple races is you can begin to dissect the race specificity. And so within this population, we found a second gene that was effective to QFCS, and both of these genes that we temporarily designate as SRTM4 and SRTM5 are effective to UG99. And you can see the additive effect of having both of the genes to race QFCSC. We were able to map the resistance conferred by SRTM4 on chromosome 2AL, which is useful for bringing that gene into wheat. And so that's kind of a taste of the very standard work that we've done for many of these genes in the wild relatives, and kind of a summary of what we found overall for Agelapis tauschii and Triticum monococcum is displayed here. And so for many of the known genes, we prioritize those for fine mapping and even cloning. And then recently in Science, it was reported SR33 and SR35, both of those resistance genes being cloned. And the collaborators for those works are displayed here. So in Triticum monococcum, we found a couple of new genes, and in Agelapis tauschii, we found 3 new genes. And all of the work, or all the phenotyping was done in our lab, and we we've made a lot of progress very rapidly by having a lot of people working on the genetics and high throughput phenotyping. One thing I'd like to point out at this point is that for the SR35 cloning project in particular, having this really strong data set of infection type patterns and knowing out of 1,000 triticum monococcum accessions which lines have SR35 and which lines don't have SR35 is really useful for cloning these genes, because the map-based cloning really narrowed down the segment to just 9 resistance genes in a tandem array, which is quite difficult to sort those out. But going into our accessions and resequencing those genes allowed us to rapidly narrow it down from 9 genes to simply 1 gene. And so having really strong phenotyping with multiple races has been quite useful. That's kind of a summary of the work from those diploid wild relatives that we've done. I'm going to switch gears a bit here and talk about characterization of adult plant resistance in a population that we've been working with. And so the line Thatcher wheat is known to have a small amount of adult plant resistance to stem rust. And so adult plant resistance simply means resistance that's not effective at the seedling stage. And we had available a population of 160 rills with 618 molecular markers, and we phenotyped that population for 3 seasons in some different environments. And what's really interesting is that if you look at these 2 parents, they're both relatively susceptible to UG99, but within the population there was quite a bit of transgressive segregation where we saw some quite resistant progeny, which was very interesting to us, because this is what you might expect for adult plant resistance conferred by quantitative minor genes. And so after screening the population in the field, we also looked at the seedling infection types to a number of races. And what I'd like to highlight in this chart showing the seedling infection types to the parents is that for TTKSK, both of the parents are susceptible at the seedling stage. But there are some races that show differences to the parents. And so we evaluated the population to both TTKSK and also to this race SCCSC just to kind of see what's going on. We found that race SCCSC had a single major gene, and that all of the population was susceptible to race TTKSK. And it's already known that Thatcher possesses the resistance gene SR12, and we assume that SR12 was the gene giving this resistance in Thatcher. The next step that we looked at was the composite interval mapping of the resistance of the adult plant resistance. And what's really interesting is this is the biggest quantitative resistance locus that we found, and it co-segregates with this SR12 phenotypic marker. So we included the SR12 qualitative data in the mapping as part of the map, and it co-segregated perfectly with the peak of this adult plant resistance QTL. So even though we don't see any effect of SR12 at the seedling stage in either of these in the population to UG99, when you look at UG99 in the field, it looks like this defeated resistance gene may actually be having an effect. Here's just a chart showing another resistance locus on chromosome 1A that we found. And there was a couple other QTL that we identified. And what I'm going to discuss next is the interactions among these different QTL. And so this is just the results of an ANOVA, and you can see the effects here. So you see the 4 QTL that we identified, SR12 and chromosome 1A. Also, we found an effect consistent with LR34 and 1 on chromosome 2BS. And then here are the 2A interactions displayed here. And the p-values and also the percent of the variation that was explained by these various effects. You can see that a lot of the 2A interactions displayed a significant amount of the variation in 2008, and then also in the other years, and the overall severity variation was explained not only by these 4 QTLs, but by their interactions. And Mark Sorrells, in his work with cement germplasm, has found that these epistatic interactions can have a significant effect as well. And when you look at what's going on in this epistasis, I think it's kind of interesting. And in this graph, I'm going to spend some time to explain, and so hopefully it'll be clear. So if you look at this graph, there are 4 different means that are displayed. The 4 means are just combinations of the effects of 2 different loci. They each have 2 alleles. And so if you look at the blue box here, this is the average disease severity of lines in the population that have the McNeil allele for 1A and the McNeil allele for SR12. So they don't have the 1A and they QTL, and they don't have SR12, and they're quite susceptible. If you look at the 1A effect alone, which is in the red box, you don't see any significant difference. So the QTL and 1A by itself does absolutely nothing. When you look at the effect of just SR12 alone, which is displayed here, you can see a slight reduction in disease severity. But when you have both of these QTL together, you get a more dramatic reduction in disease severity. And so if you're familiar with epistasis, the fact that these 2 lines are not parallel is indicative of epistasis, not just quantitative resistance. And so when you look across all of those different 2A interactions for the different QTL, you see it's very common to have that same pattern of where when you have both alleles, you see increased resistance and not just an effect conferred by one. The exception being this interaction here. So that's just kind of an overview. When you look at the effects of different combinations across the population, you see something that's even more interesting. And so in this bar chart, you have the averages of different combinations of QTL, which are displayed here. And it's a little bit difficult to read, but I'd like to highlight that the SR12 and 1A QTL were very key in providing resistance. And so this combination is in column 6, and you can see a reduction in disease severity. But if you look at the 4 QTL by themselves, in these 4 columns, you see that there's essentially no effect of the QTL alone. And to get a really reduced level of disease, you need several of these QTL together. Unfortunately, since we're talking about combinations of QTL, for some of the categories, we don't have enough lines to come up with a mean. And it's really unfortunate because we only had 1 line that had all 4 of these QTL together, and it was quite resistant. However, there was more than 1 line that was very resistant. And so when you look at those lines carefully, we saw that they had recombination within the QTL, so we weren't able to include those in the means. And so here's 4 of the very resistant lines, their mean severities in different environments. And then you can see that for the 2BS QTL, 3 of them had recombination within the markers flanking that QTL. So we weren't able to include those in that very resistant category because we don't know for sure if they have this 2BS QTL. But certainly we can see that the other 3 QTL are conferring some disease resistance. So this epistasis, I think, is kind of unique. Usually you don't see this strong of epistasis for any trait. And so I think it's very interesting that we see this for adult plant resistance in stem rust. And then the last really component of the research that I'm going to discuss today is the dissection of resistance on chromosome arm 2BL. And so I went through some information on wild relative resistance and also adult plant resistance. In this section, I'm going to talk about some major resistance genes on chromosome 2BL. We started with 2BL when we were mapping resistance in this line called GABO 56. And you can see that this line is quite resistant in Kenya compared to susceptible Chinese spring. And when evaluating the progeny of a population at the seedling stage, we found evidence of a single major gene that we temporarily designated as SR GABO 56. And we mapped this gene to chromosome arm 2BL. And what's significant about the map location here is that it's at the same location as some of the SR9 alleles that have been described. So SR9 has currently 5 different alleles that are effective to stem rest. And none of those 5 previous alleles were effective to UG99, but this is potentially an allele that is effective to UG99. And so we kind of prioritized this resistance gene because I think it's a nice system that if we can clone it, we can really look at what can explain all of this phenotypic variation at a certain locus, which could be interesting for understanding host pathogen interactions. So when you go to chromosome arm 2BL on the map from what was previously known, you see that also SR28 is there. And so we wanted to know if potentially we made a mistake and this is SR28, or maybe how far away the Gabo56 gene is from SR28. So I made the cross between a line carrying SR28 and also Gabo56 and evaluated the progeny and came up with the finding that they're about 20 centimorgans away, which is consistent with the map location. So we decided also to map SR28 and indeed found that it's about 20 centimorgans away when you comparatively look at the maps. And for the SR28 story, we found that the closest marker was actually a DART marker, and so we converted that to a PCR-based marker and evaluated the flanking markers within Minnesota, North Dakota, South Dakota germplasm to see if it might be useful for marker-assisted selection. And in fact, it was the combination of those 2 alleles was diagnostic within that germplasm. And the last point that I'd like to make regarding chromosome arm 2BL is in part of the TCAP project, we're evaluating around 5,000 different accessions of wheat as a core collection within the germplasm bank. And when we did an association analysis between the 9,000 SNP markers available at the time and resistance in that germplasm, we found about 35 markers that were significantly associated with UG99 resistance. What's very interesting is that all of those 35 markers were on chromosome 2B, some on 2BS, some on 2BL. And so the chromosome 2B is really very interesting for UG99 resistance. And within that data set, we're able to postulate which lines carry SR28 and which lines carry Gabo56 based off of the screening with multiple races. And we ended up finding a perfect marker within that germplasm for SR28. So we're currently working on expanding the fine mapping in SR28, going from that marker and moving on. So it's kind of exciting to have those resources available. And to kind of summarize some of the work that we've done, this is just, again, that slide showing the status of where we were in 2008. And then by working with a lot of people from around the globe, in just 5 years, the community has added a large number of resistance genes in a short matter of time. And so the number of resistance genes has just about doubled in about 5 years. And so shown here are many of the green QTLs that Mark Sorrells and Ravi Singh have identified. And additionally, you can see a lot of major genes that have also been added. So just comparing these 2 cartoons, you can really see that there's been a lot of progress. And for the major genes, almost all of the phenotyping was done in our lab. So in summary, I hope that I've answered the question, what is UG99? That you can go away and know that it's an isolate of Buccinia graminis that was found in Uganda in 1998. And it's a threat to wheat production, and it's not a simple race, it's a complex of races. And I hope that you have an idea of some of the work that we've done. It's very basic work, but given the threat of UG99, we've kind of been under pressure to make a lot of progress in finding a lot of genes really fast, and I think we've been successful in doing that. I'd like to acknowledge a lot of people, particularly my lab, and then also Eugen lab and Les Sabo's lab, who are also scientists at the USDA Cereal Disease Lab. In my lab, there's a few people I'd like to highlight. Jayaveer Muthu Nirmala, who's a postdoc, who's really an excellent scientist. She has a couple of first author publications in PNAS, and then Jordan Briggs and Josh Kielsmeyer-Cook, graduate students. The field phenotyping would not be possible without work with Kenyan and Ethiopian collaborators, also the University of Minnesota, allowing us to work in the biocontainment facility. A number of collaborating labs I've listed here, which we've worked with in the mapping of all of that doubling of resistance genes. So Stephen Zhu and Mike Bondman from the USDA, Ravi Singh at CIMMYT, a number of labs at Kansas State, George Dubkovsky at UC Davis, the Pumphrey Lab at Washington State, Hebert Lab in Canada, Mark Sorrells' lab. Some sources of funding, of course, the DRRW, the Pakistan Wheat Production Enhancement Project, and the National Plant Disease Recovery System and the TCAP project. And with that, I'd like to take any questions. Thanks.